Fine. In which what we have taken up, these test tubes in which there is a growth of the bacteria. Okay, basically you call it as bacterial inoculum, which you put it into the molar Hinton broth. First thing, fine. And because of this growth, as such, there was a turbidity in these particular test tubes. There was a turbidity, I'm telling, okay, because of this particular growth, which was same everywhere before. But what you have done, you have added up this particular antibiotics to it. Okay, in the decreasing gradient or increasing gradient, from any one side you can pick it up. So suppose we pick it up from this antibiotic concentrations highest one. So in the first test tube you put up what? 128. Then you decrease it to half, 64. Then 32. Then 16. Then 8. Then 4. Then 2. Then 1. 0.5 and 0.25 microgram. This is the drug which you have put up into these particular broth which you have taken in that particular test tube of a particular bacteria. Now you want to find out the MIC and MBC of this. Fine. So this is the antibiotic concentrations per ml which you have put up in the micrograms, and you want to calculate up the MIC and MBC. Now, when you come next day, this is a picture which you find next day. Fine. What you have observed, that the concentration where is 0.25, it remains turbid. That particular broth which was like day before like having a turbidity and all, still it is being maintained up, and in fact that is like same or increased up. Then 0.5, that is again turbid. 1 microgram again turbid. 2 microgram turbid. 4 microgram turbid. So up to 4 microgram this turbidity is maintained up, which you have like inoculated up a day before. Then at the 8 microgram, as you can see that there is a clarity in the broth. Why this clarity is there? This particular clarity have come because at this concentration of drug, that is a 8 microgram, there is an inhibition of the organism. Okay, there is an inhibition of this bacterial organism, and that particular thing is it like depicted about by this clarity. Fine. So what you do, you take the like loop and you put it over the culture plate. Fine. You take a swab into this and you just put it over this particular culture plate. What you observe, that growth happened. So why this growth happened? This growth happened because bacteria is still there. Fine. Bacteria is not being killed fully. It is still there, but the inhibition of the growth have happened up. And that inhibition of the growth is determined by what? This clarity which you are observing up into this test tube. Fine. Now at 8 microgram the clarity is there, at 16 microgram also the clarity is there, but they are growing up on the subcultures on the culture plate. That means 8 microgram, which is the lowest concentration at which the clarity comes, that's why it is termed as what? MIC. That is the MIC of this particular picture or this particular test is what? 8 microgram per ml. I suppose you got my point. Then you have what is MBC. MBC is that particular thing, that highest dilution of the antibiotic, which shows 99% of the inhibition of the organism, or 99.9% some says, and that is same for the bacteria, and that is also there for the fungus also. We use that. Okay, so basically we are talking about bacteria here, to MBC, minimum bactericidal concentration. So you can see up that at 16 also they grown, but at 32 there is a clarity and there is no growth also on the subculture. That means now the full inhibition of the organism, they are being killed off 99%. So that is what is the MBC of it. So what you have got from this example, that the MBC here is more than MIC. Okay, so what is MIC? That is concentration at which clarity comes, but still the bacteria is there. But this particular concentration 32, all the bacteria have been killed, mostly like 99% or 99.9%. Got my point? This is the way, this is termed as broth dilution method, by which clearly you can find out the MIC and all. Okay, so this is what is termed as broth dilution method. Okay, other than that, you remember an important test nowadays have come, that is termed as test or absilometer test. E-test or absilometer test, and this E-test or absilometer test is having the antibiotic gradient here, and this particular antibiotic gradient you can see up by this particular zone here. Okay, so as you can see that this particular zone is like converging on which particular number, 0.25. So what is the MIC of it? 0.25 microgram per ml. Okay, so that is being calculated by this particular method termed as what? Absilometer test. So this is one absilometer test. One is broad dilution method by which you can calculate up the MIC. There is one more which is termed as automated systems, and these are what? Vitek 2, Phoenix and Microscan. So these are the newer ones which have come. That is the automated systems, Vitek 2, Phoenix and Microscan. And what you do in them, you just prepare a solution, okay, or the required concentration and all, 0.5 microfarad standards and all, and you just put up in that into the this particular machine, and machine will calculate up the MIC of the various drugs against that particular organism. Okay, so that is what is given by these automated systems. So the question of ours was like, which of the following does not determine up the MIC? So answer is what? MIC is not determined by which method? Kirby-Boyer disk diffusion method. Okay, now this Kirby-Boyer disk diffusion method, I will make a separate video for it, but just for the instance I will show you this picture. This is what is Kirby-Boyer disk diffusion testing, that is KBDT. Okay, disk diffusion testing also they say, so you can remember KBDT. So this is what is termed as Kirby-Boyer disk diffusion method, and in this you already know that you prepare a lawn culture and then you put the antibiotics and all, and around that antibiotic there is a zone of inhibition which comes up, which suggests up that this particular antibiotic is working on this particular organism. So this is what is like you do for antibiotic sensitivity testing, but this is not used for what? This is not used for MIC. So I suppose I made myself clear that you have e-test nowadays on which the question can come. Broad dilution method is there. The automated machines are there now, that is termed as Vitek 2 and Phoenix and all. But this particular method, Kirby-Boyer disk diffusion, is not used for the MIC. And what is MIC? Minimum inhibitory concentration. And what is MIC? As per the broad dilution, at which you could be able to see of what? The clarity in the test tube. But when you subculture it, the growth comes on the culture plate. Whereas in the case of MBC, when you subculture, nothing comes. So in example, you can see the 8 microgram per ml was the MIC of ours, and 32 was the MBC of ours. So I suppose this particular concept is clear to now, and we'll discuss further this Kirby-Boyer distribution in a separate video. Thank you so much.